Hello viewer, a warm welcome to Elimu TV where exclusively watch and learn. I am a teacher, Madam Christine Okoth, taking you through the 9th lesson of Biology Form 4, and today we're going to learn about gene mutation. We're still under the same topic, the first topic in Form 4, which is Genetics 2. Welcome. The second question: what is the difference between gene mutation and chromosomal mutation? So chromosomal mutation, it is any change in the structure or the arrangement of the chromosome. So any change that is in the structure or the arrangement of the chromosome, that one is defined as a chromosomal mutation. Whereas the gene mutation, it is a permanent change in the DNA sequence that makes up a gene. A small recap of where a gene is located, where DNA is located, and where the chromosome is. If you remember the structure of a cell, we have this part we call the nucleus. Inside the nucleus, we find what we call the 23 pairs of chromosomes. Then on the chromosomes, that is where we find the genes, and the genes they consist of a DNA. So it is a sequence. From the nucleus, you get the chromosomes. From the chromosomes, you get the gene, and the genes are made up of the structure that we call the DNA structure. So today we're going to learn about gene mutation. So what exactly is a gene mutation? This is the change in the structure of the gene, then which it results to when there is alteration of the sequence of the gene which is forming, that is whereby it leads to a mutation. So this is a permanent alteration. Mark the word, it is a permanent alteration of the gene, that is the DNA sequence that makes up a gene. So the sequence differs from what is found in most people. So mutation will mean that the sequence of your DNA is different from what is found in other people. So the change in one of the bases or the multiple base is what leads to the gene mutation. So we discussed earlier that we have 4 bases which are found on the structure of the DNA. So if there is any alteration or if there is any change in the bases, it can either be one of them or it can be multiple bases. So that one will obviously lead to what we call a gene mutation. Structure of them means by a gene being deleted from that what it was intended, like in our example, completely different. Another type of gene mutation, we have insertion. So this is where, just from the word, it has been inserted. So this is where an extra unit will be added in the genetic sequence. That unit was not there, so it has been inserted. So it completely alters the meaning of the word. For example, we have the word eating, E-A-T-I-N-G. If you add a H in the beginning, the meaning will be totally different. It will be eating. Eating and heating are 2 complete different words. So you can see from our illustration here, trying to explain, there are some bases, that is adenine and thymine, have been added in that sequence. They were initially not there, but they have been added. So due to the purpose that they have been added, there is completely change of the sequence of the gene. It will be no longer the same, so it will have another different meaning. Another type of completely of a gene sequence is double gene of the gene sequence. So we have different types of gene mutation. Number 1, we have albinism. We have the sickle cell anemia. We have the Haemophilia, and we have the color blindness. We're going to look at each of them and define them. Some of these we have discussed in the previous lesson. For example, the Haemophilia and the color blindness, when we're talking about the genes which are linked to the chromosomes. So the first one is albinism. You've all heard about albinism. So this is a condition whereby the skin of an individual will fail to produce the skin pigment which is known as melanin. So if a skin does not have that pigment we call the melanin, the condition of the person, you say that that person will be having albinism. If you look at our illustration, there's somebody who has black hands, there's somebody, so that means that this person has enough pigment in the skin. So this other person who is light skin or the pigment is very different. That means that he does have very little melanin in him. So that is a condition we call the albinism. So melanin is the one that gives the skin its true color. So it also protects the skin and the eyes against the harmful rays. So if you look at the mutation, if A, we can say in albinism, A is the dominant gene. So it means that it will influence the production of a melanin. If you have the small a, that is the recessive gene, which means it prevents the formation of melanin. So if you have this recessive gene, it means that you will be an albino. So let us look at the genotypes and the phenotypes. Genotype is the genetic makeup of an organism, physical appearance of an organism. So if somebody has a homozygous dominant gene of albinism, it means that that person is pigmented or is not affected by albinism. So he will have all capital A. So those genes are all of them dominant. Heterozygous, which is different, it means this person has the genotype will show a capital A and a small a. So on the genotype you cannot know, but seen on the genotype. Then if you look at the last type of genotype, we have the homozygous recessive. So this is a person who has no pigment. So this is the albino. He has the recessive type, that is all of them the small a. So these are the signs and the symptoms of a person who has albino. So number one, they lack a skin pigmentation, which makes more susceptible to sunburn and the skin cancer. So if this person does not have this melanin, it means that he will be affected by the rays of the sun. Then there is an absence of the normal pigment in the eye, resulting to they either have a very pale blue or the red eyes. So if you look at the illustration, we have two boys here who are albinos and the black boy, that one is just fine. So you can look at some of the signs which you'll identify that a person has albino. Another effect of gene mutation is in sickle cell anemia. So this is a condition whereby the red blood cell, they have an abnormal shape or a sickle cell shape. This will always lead to the inability of the blood to transport oxygen to all parts of the body. So looking at our illustration, we have the normal red blood cell. It is like disc-like in shape, whereas a defected red blood cell will be sickle-celled, like the one which is showing in our illustration. So these are the alleles and the genotypes and the phenotypes of a person which are possible of a person who is having sickle cell anemia. So we have the dominant allele, that means it is responsible for the dominant gene. So we have the genotype, which is HBA and HBA. That shows that this person on the phenotype, this one is a normal person. Then we have the recessive. So the HBS, that one is the one which is responsible for the defective red blood cell. So that is the HBS. So if the HBA and the HBS, they will combine, it will have a phenotype that is known as the sickle cell trait. This person cannot show when you look at the person, but the genetic makeup shows that there is a defect in the red blood cell. Then lastly, we have the HBS and the HBS. This shows that this person, all the red blood cells are defected, and this person, we say that is sickle cell anemic. So we've said that hemophilia is the inability of blood to clot, leading to severe bleeding. So if you've ever seen somebody, if you just cut or you injure a blood vessel, this person will continuously ooze blood out of him. So this person, there's a defect in the clotting of the blood. So this person is said to be hemophilic. So in our illustration, it is showing if somebody blood vessel, then there is a damage in the blood vessel, the vessel will clot very fast. So it will prevent the oozing of the blood. So this person is healthy. Then if this person had a normal blood vessel, then in case the blood vessel is injured, the blood, the coating is So if this person has normal blood vessel, then a vessel is damaged. The inability to clot, and the person will be losing so much blood. So this condition is what we call as hemophilia. So let us look at the genotype and the phenotype. So in male, there are only two possible phenotypes. So this person, if it's a normal male, it is denoted as X, H, and Y, because it is a sex-linked gene and it's normally on the X chromosome. Then, if this person is hemophilic, it will be denoted by a small h on the X chromosome. Then, on the female, we have three types. So we have the normal female, it will be denoted by the X and the capital H to show that this person is normal. Then, we have the carrier. This means that he has both of the alleles, the capital and the small one. Then lastly, we have a female who is affected, a female who is hemophilic. He will have both the small h denoting that the female is hemophilic. Then another effect of gene mutation, that is in the red-green color blindness. So this is a disorder which is caused by the mutation of the normal color vision. So this person has a disorder, especially when he's trying to see images which are either green or red, or any shades of green and red. For example, if it's a green apple, this person will tend to see something which is brownish or something. The pigment cannot detect the color green. If this person was looking at an apple which is red, he will not be in position to look at, to see that it is red, but rather he will see something different which is different from the red, maybe something like a brown. So any shade of green, any shade of red, or the real colors green and red, this person is not in position to see these colors. So thank you very much for watching. Remember, you can write to us. You can also write any feedback, any question, any clarification you'd like to have known. So our Facebook page is Elimu TV. You can write to us. Our Twitter handle is at Elimu_KE, and also keep the SMSs coming. Our SMS number is 225